I'm going to hand over to Alistair Smith and allow him to talk you through the investment opportunity. Alistair.
Thanks, James. Thanks very much. Thanks for the opportunity to present, and good evening, ladies and gentlemen. So I promise I will not make it complex. I'm going to try and keep it very non-technical as I can. Happy to go through more of the detailed science afterwards, but I think we'll keep it as high level as possible in the presentation. Okay, so I thought, in the interest of keeping it non-technical, I would start with some key investment messages. And I think the first important message to get across is we are a preclinical biotech, but we are not a single asset or two assets biotech. We have a platform technology. So we've developed a drug development platform, which we call the Affimer technology. And it's a platform technology capable of delivering numerous drugs into our own development pipeline and also to partners. So there really is no limit to the number of potential drugs we can put into our own pipeline and into partners' pipelines. That's large partners such as the American biotech Moderna, and smaller partners, we can talk more about those later on. But it's not a binary one or two assets win or lose type of biotech risk. Secondly, the area that we are working in, the area we're focused in drug development is immuno-oncology. I'll say a little bit more about that as we go through, because it is such a hot area. As I put the figure up there, over $20 billion of deals, M&A licensing over the last couple of years. There is a big one in there actually, there's a 12 billion acquisition in there, so it's about 11 or 12 billion of other deals, other licensing deals done over the last couple of years. So it's a very hot area, and a lot of those deals are preclinical. Okay, you don't see that in many disease areas. A lot of the deals are done at quite late clinical stage. So for an early stage biotech like Avacta, focusing on immuno-oncology gives you the chance to do some really quite significant deals at quite an early stage of development. You don't have to wait to be in phase 2, phase 3 to do those deals. So we're focused on a very hot area. We're also generating revenues, even at this early stage. So the platform, as I'll describe it to you, the Affimer platform, our focus is on drug development, but we're also commercialising the Affimer technology in diagnostics and other non-therapeutic areas, so generating revenue straight away. Through the same licensing opportunities, the values of those licenses are smaller, but the key point here is that the technology is being used globally by a wide range of diagnostic companies, academics, drug companies, so the technology fundamentally works. We've made hundreds of Affimers, partners have used many of these Affimers, the technology works. We are well beyond any question of whether the technology works or not. And so the final message is that combination of non-therapeutic, revenue generating, building towards a profitable business unit, selling reagents into those non-therapeutic areas, gives us a very low downside risk. We are commercialising the technology now, so the downside risk is very, very low. Whereas the upside, if you think about some of the comparators, and we'll talk about these later, the upside valuation of clinical stage assets like Affimers is very, very high indeed. This company here, just as an interesting comparator, some of you may know this company is AIM listed by Aventix. They have a comparable technology working entirely in the diagnostic area, and you can already see that a profitable, quite a small 7 million turnover business, but profitable, their valuation is significantly greater than ours. So we sit between these two areas, and as you see, my argument would be we are hugely undervalued at the moment for the technology that we've got. So those are the four key investment messages. We're a company of about 80 people. We're split over two sites. The drug development is carried out in Cambridge. The non-therapeutic diagnostics development carried out in Wetherby in Yorkshire. We have a strong balance sheet. We have a very good set of shareholders who are extremely supportive. And our push is to build that diagnostic business towards profitability and to get the first Affimers into the clinic where there's that significant value inflection point. Okay, so I've mentioned it several times. What is an Affimer? And without, again, getting too technical, what you need to understand is why antibodies have any real commercial value. And the reason is that an antibody can bind a target. So just think about your immune system where antibodies are raised to bind to a virus or a bacterium to get the rest of your immune system to deal with the infection. This property of being able to bind to a target, folks have harnessed that over the last 20, 30 years and started to get antibodies to do other things. So for example, if you can get the antibody to bind to a drug target and block that drug target, you have the potential to create a therapeutic. A third of all drugs in development now are monoclonal antibodies that do exactly this. Okay, so it's the ability of an antibody to grab hold, capture a target, and block it or do something with it. That's why antibodies have around $100 billion worth of market currently. The problem is they're big, they're difficult to develop, they're very costly to develop and manufacture, they're relatively feeble, they're not particularly robust, so in many applications they struggle because they're damaged in the applications. So finding a small, simple, robust alternative to antibodies has been a push for about 20 years now. And to cut a long story short, what we have done is develop a very simple, much smaller, very robust, chemically, thermally and biologically alternative that does exactly the same thing as an antibody, binds to a target very specifically and can then do things with that target, and we call it the Avacta technology. So if you want to think about a faster, better, quicker antibody, that's a good way to look at it. So it's entirely proprietary to Avacta, so we own all the IP around the Avacta technology. Nobody else can develop an Avacta without licensing our background IP. It's not immunogenic. Okay, so that means it doesn't elicit any sort of immune response in human samples, so there's no real strong likelihood of issues when we get into the clinic, at least from the core technology perspective. We can generate very good Avactas, so I can go through the technical details later, but we can generate very good Avactas, as good as most monoclonal antibodies, in a few weeks. It takes many, many months to a year to generate new high-quality antibodies. We have a process now that generates new Avactas in a matter of weeks, 12 weeks or so. They are very specific to the target of interest. So when we create an Avacta to a new target, let's say that's a drug target, we can make sure that that does not cross-react with other targets. That's not often the case with antibodies. Quite often they cross-react with other targets. And of course, cross-reacting means you get off-target effects in therapeutics, side effects which you don't want. In a diagnostic, it might mean you get a false positive. So being very specific to the target and nothing else is a really important property. Okay, so what we have done over the last 2 to 3 years is develop a business model that's split into two. So as I said, we're developing therapeutics. I'll talk a lot more about that, and I won't focus too much on the reagents, but especially in this area called immuno-oncology, which I will explain. That's a standard biotech business story. So it's going to take many years to get through into the clinic to partner those assets, but when you partner them, there are some really significant values attached to it. The reagents business is a much nearer term. We're commercialising now, generating revenues. This we should be able to push through into profitability in the next 2 to 3 years. And this is all about providing Avacta reagents to replace or complement antibodies in diagnostic and other laboratory products. We'll say a little bit about that later on. So there are two themes to the business strategy. Let's focus on the therapeutics to begin with. And I've mentioned immuno-oncology a number of times, and it needs a little bit of an explanation. So what you need to understand is the immune system, your own immune system, is perfectly capable of dealing with cancer like it does with any other infection. But there are things that the cancer has evolved to do to evade the immune system, essentially. So your immune system does not see cancer as a foreign thing. So it passes over it and doesn't attack it. So if you give the immune system a little bit of help so that it can recognise the cancer as a foreign thing, then it has all the tools that it needs to be able to attack the cancer and in principle give long-term benefit, even cure. And there is, if you go on Google and look on the internet, you'll see a huge amount of literature and stories and some fantastic clinical successes that have been achieved with immuno-oncology in the last 10 years or so. So it's all about harnessing the immune system to fight cancer. So without going into all of the detail on here, what I thought would be useful would be just to say, just to pick out some key words that you'll hear over and over again in presentations like mine and other companies. I think you need to understand what these are, otherwise obviously it gets a little bit lost very quickly. So there are 3 things you want to do in immuno-oncology with a technology like Affimer's. First of all, you want to be able to stimulate the immune system to attack the cancer. And the phrase that we use there is agonism or agonists. So if you hear about agonism, it's about stimulating the immune system to attack the cancer. You also want to be able to block certain signals. So cancers have evolved to display on the cell surface signals to tell the immune system, leave me alone, I'm a perfectly healthy cell, don't attack me. Those signals, if you can block them and switch them off, the cancer reappears as an aberrant cell and the immune system can attack it. So these are signals that the cancer cells are using to fool the immune system, and you would like to block those. And blocking we refer to as antagonism. So agonism is stimulating, antagonism is blocking. So those are the first 2 things you want to be able to do, both of which we can do with Affimer's. And the third thing is you'd like to be able to target things to a tumour. I'll give you one very, very simple example. Chemotherapy, so chemical toxins that are given to patients, that are given systemically, go around your whole body, lots of side effects that kill tumours. If you can target the chemotherapy, for example, by attaching the chemotherapy to an Affimer that takes it to the tumour, you get much more directed therapy of the tumour and lower side effects. So targeting other types of therapy to the tumour is the third area that large pharmaceutical companies are all trying to achieve these different things. And without going into a lot of detail, the slide here is split into 2. On the left-hand side is the work that we are doing in our own drug pipeline in-house. And the key benefit of Affimer's that address those agonism, antagonism and T-cell engagers are the fact that we can make multimers. So we can stick Affimer's together in 2s or 3s or 4s. That's extremely difficult to do with other technologies, particularly antibodies. The fact that we can do this allows us to look at agonism and antagonism and T-cell engagement without going into the technical details. If you can't do this, then you can't address those types of therapeutics. So we focused our in-house pipeline entirely in those areas. Something called T-cell engagement, which I'll talk through afterwards if anyone's interested, making inhibitors, multi-specific inhibitors and agonists. If you can't make these structures, you can't address these sorts of therapeutics, and we can make them particularly well. The other key technical benefit of Affimer's is the size and the simplicity. They are 10 times smaller than an antibody, very, very simple in structure, which means that cells in your body can make Affimer's really easily. The small size and stability and ease of production means you can start to look at some other very, very high-value therapeutic opportunities, and we're doing these with partners. We simply don't have the resource to do all of these things in-house, but we're doing these with partners. For example, gene delivery with Moderna, which is a very large US biotech, very, very well funded, as well as other smaller partners. Working on CAR-T therapies, which have had some enormous success in recent times. Kite, one of the leaders, was just bought for 12 billion by Gilead. These are very hot areas. And drug conjugates that I just mentioned, where you target the chemotherapy to the tumour by combining chemotherapy and something like an Affimer. These are all things we're doing effectively off our balance sheet with partners by providing Affimers that they are interested in to particular drug targets and allowing them to get on with it, which provides us with the proof of concept data to then go out and do business development and get further licensing deals. So in-house we are focused on making these multimer structures, dimers, trimers, tetramers, and that really sets us out as unique in the field. I thought I would just give you one example. I know James likes this particular example, but it illustrates one particular use of these multimers. Okay, so there is a There is an area of oncology of cancer therapy called T-cell recruitment. What you do with T-cell recruitment is you try to make something that goes and gets your T-cell, your immune cell, and attaches it to the tumour. Okay? To engage the T-cell, to engage your immune system with the tumour. In order to do that, you need something that's got an end that's sticky for the T-cell and an end that's sticky for the tumour, in simple terms. And so what happens is the dimer, the Affimer dimer, is put into the body, it connects with the T-cell, connects with the tumour, sticks them together and kills the tumour. There is already a marketed drug that does this called Blincyto, made by Amgen. It is one of the most, I think it was the most expensive oncology therapy. It's not marketed in the UK because of cost. NICE don't approve it in the UK, but it is very, very effective in many forms of cancer, particularly leukemia. In order to be able to do this, you need to stick two things together, one that attaches to the T-cell and one that attaches to the tumour. So what we could do is we could make an Affimer equivalent of this that looks just like Blincyto and compete on cost, because Affimers are much, much cheaper to produce than antibodies. I don't think that's the right thing to do. I don't think that's strategically the right thing for a company of our size to do. More interesting is to take the next step, because once you start to attack a tumour through a particular biomarker on the tumour surface, then you'll kill the cells that have that biomarker, but the tumour will grow with other cells that don't have that biomarker. So the tumour escapes that therapy. So what you would like to be able to do is attack two things on a tumour to prevent that tumour escape. So straight away you need to be able to stick three of these things together. So far that's not been done with antibody technology. And I should say, although not in this particular application, we've made trimers and tetramers and shown that they work with Affimers. So in principle this is absolutely doable with the Affimer technology. So what happens is very similar, but this time when you attach the T-cell to the tumour, if the tumour starts to grow other types of cells that aren't rich in the first target, you can still attack it with the second target. So that's already a novel therapy that prevents tumour escape. But the problem with all of these technologies is they're quite small, so they get cleared out of the bloodstream through the kidneys into the urine quite quickly. So you'd like to be able to keep the therapy in the bloodstream for a lot longer. Blincyto, for example, is delivered to the patient on a continual infusion over hours and hours, rather like chemotherapy, because it's cleared from the blood so quickly. So what we've done is we've developed an Affimer that binds to one of the really big proteins in your blood, so that it piggybacks the whole thing onto one of your very large proteins in the blood, and therefore stays in the blood because that big protein is not cleared from the blood. So effectively it's grabbing hold of something large that can't get cleared through the kidneys. It now spends a lot of time in the blood, many, many days, up to weeks, and that allows the thing to have a long time in the bloodstream, lots of exposures to the tumour. But the principle is exactly the same. Ultimately, it recruits a T-cell, engages it with the tumour cell, and kills the tumour. Now that would be a highly differentiated, very valuable oncology therapy. Okay, that's just one example then of why you need to be able to build these bigger structures of dimers, trimers and tetramers to really have any significant value in these types of markets. And I said I would mention the comparators. Molecular Partners, Pieris and Ablinks are all clinical stage. So I think that's important to make that point. They're all clinical stage, so 2, 3, 4 years further ahead than we are. But look at the sorts of valuations those clinical stage companies achieve. We're expecting to go into the clinic 2019, 2020. And these are all 10x, 20x, 30x our current valuation. See, James is hovering, so I'm going to speed up. The second part of the business that is generating revenues right now, just run through very quickly, this is the use of Affimers in kits for doing experiments, so non-therapeutic, very few regulatory hurdles, being used in laboratories, in diagnostics, for example, pregnancy-style, rapid testing. Affimers have been proven in all of these applications, and we have evaluations ongoing with 20 to 30 diagnostic and other companies now who are looking at taking the Affimer technology into their product development pipeline to replace antibodies. Very quickly through the figures, more than 50 projects done with those types of companies over the last few years. Order book up considerably over the last year, nearly doubled over the last year, and we did the first significant licensing deal in 2017 with one of the big global diagnostic companies, Siemens, Roche or Abbott. We're not allowed to say which one, but one of those top 3 diagnostic companies who took an Affimer into their diagnostic development pipeline, and there's multiple evaluations of that sort of nature ongoing at the moment. Obviously we can't talk about those, but it's an exciting time. I think 2018 will be a very exciting year for the company. Very briefly, the management team, talk to me later on, but just to highlight, Matt Johnson was the head of R&D at Abcam, joined us a few years ago, responsible for developing the technology and for operations on the diagnostic side. Amrit Basran was head of biopharm formulation for GSK, came into GSK through an acquisition of a similar type of technology a few years ago. And Philippe Cottrell was the chief commercial officer of Abcam and joined us about 18 months ago and has been responsible for driving that revenue growth on the non-therapeutic side. Very, very briefly, the 2016-17 was a very, very good period for us. We de-risked the technology enormously. We made a lot of progress. We filled the pipeline of collaborations and potentially licenses from evaluations. What you really want to understand is what are the milestones, what you should be looking at over the next 2 to 3 years to really measure performance of the company and progress with the technology. I'll probably just highlight these 3 things. The technical progress clearly is difficult to understand. It's difficult for you unless you have the right background to ascribe any sort of value to technical progress. But the key commercial steps are licensing deals, both the non-therapeutic and the therapeutic licensing deals, which I think we have a very good chance of delivering in 2018 and certainly onwards from there. And the other big value inflection point, 2019 to 2020, is first-in-man clinical trials, as we take our first Affimers into human trials. In summary then, the Affimer technology absolutely works, so we are well beyond any question of whether we have a working technology or not. We're very confident we can now develop Affimer therapeutics into the clinic with this platform technology. We have very strong and broad IP around the Affimer technology, so we're very confident of the IP position. We have this dual business model generating revenues. Ultimately, I think these are 2 separate businesses. I think there's a share for share and 2 separate businesses in due course. I would say there is very low downside risk to the current market cap and the revenue generating potential of the non-therapeutic side, with a really significant upside on the therapeutic opportunity in immuno-oncology. I think we have a management team. We'll add to that. We're looking for a chief medical officer as we head towards the clinic. We're looking through potential candidates for a chief medical officer at the moment. We have a very strong, strong balance sheet and a strong set of shareholders. Thank you.